No? Oh, well, depends how you want to watch it. All right. I'll just start. We have plenty of formalities, so this one is going to be less formal. Thank you very much for giving me the opportunity and to talk at this time at the meeting, where you would have heard a lot of information and a lot of interesting messages. Francesca and Alessandra asked me to reflect perhaps on what we learned over the last 10 or 11 years since we identified the cause of this very special disorder and perhaps what it all means. To all of us, it probably feels like we haven't achieved a lot, but by comparison to other diseases, other groups, I think, surprisingly also to me, this field moved dramatically forward. So I will have a few perhaps simple messages, and some of them you would have heard, others hopefully going to be new and hopefully informative for you. Let's go how we go. Doesn't want to move? Yeah. So a couple of introductory slides. As you know, the genetics is very important, and we are different for different reasons. In terms of height, it can be nutrition. You know that if you are born up in Northern Europe, you are usually taller than if you compare to people born in Southern Europe. It can be a hard work, and of course it can be a DNA. Sometimes it's a combination of all of these factors, and that's probably what we see in PCDH19. This is the space, this is when I finished my university, and this many genes and disorders have been characterized over that time. It's a lot. First epilepsy gene, which was found in Adelaide, was about 95. PCDH19 was identified about 2008, as you know. We have more than 4,200 genes almost known from about 6,500 different diseases. Just from the time PCDH19 was found, we have more than 2,500 additional genes and probably many more disorders. So the field is complex. When you look at different determinants and you do the overlap using these charts, you see that there are some genes which can cause different disorders. And when you place the PCDH19, it fits for the time being somewhere here. It's being involved in learning disabilities, epilepsies, and autisms, and there are probably 74 other genes similar like that. And we need to learn also from them. SCN2A is one of those. It's a known epilepsy, autism, developmental delay gene. And we shouldn't be forgetting to really keep in mind other examples of other disorders when we study PCDH19 epilepsy. So historically, we found this particular disorder by studying large families. And that also reminds me of an aspect which we haven't discussed very much, and that is the non-penetrance, that is up to 20% or maybe even more of individuals who carry these variants, and some of them are even in the room, who are fortunate and do not present with the complex clinical presentation as we keep hearing about your daughters or granddaughters or sons or grandsons. The first really population-based study now in Scotland estimated about this prevalence about 1 in 20,000 female live birth, which would mean that in Australia we would have about 15 cases per annum, and we don't really see all of them, and in Italy probably about 20 to 40. Christelle Depienne and her team were very fortunate when they studied Dravet-negative cases to find this mosaic male, and of course from that really the real impact of PCDH19 in early onset girls clustering epilepsy came out. So that was about 10 years ago in 2009. So these are really in nutshell numbers. We have more than 300 patients, and I would say there's well over 1,000 internationally. The onset, we know that is a drug-resistant epilepsy, quite complex, mostly focal seizures. Intellect vary. There are very severe other problems, not just seizures, and I will touch on that. The treatment is a bit of a mess at the moment, and we discussed it. I will have another perhaps slide or two to give you another piece of information. And you just heard beautiful studies, really beautiful studies about the possible mechanisms and how this mix of cells leads to the fact of the seizure, increased seizure propensity in those individuals. So with Christy, who was here 2 years ago, but now is writing up her PhD, we also looked from the perspective of increasing numbers. So initially, Christy just looked through the literature about 2 years ago, and we collected these 271 patients. There were also 12 males. Now there is more of them. They are really under-ascertained. A lot of people don't realize that males can have these kind of mutations. And we can say that this post-zygotic somatic mutation is reasonably frequent. So it's a case when this mutation doesn't come from a mom or a dad. It actually happens as an error during early development in these individuals, which we will never be able to control. We'll be able perhaps to test for it early in pregnancy, but not before. So we found as part of this study that the mean onset was about 1 year, so 12 months, while the median was about 10 months. And that agreed even with our earlier studies when we put the onset on the background of the hormonal levels, and that's how we came up. Perhaps that hormones might be involved because this is the mini puberty. There is the time when they have more hormones from also the mum, or this is the time when our bodies are prepared to enter the outside environment and they need to be protected. And then the offset was about the time of the puberty. We have seen that. Now we complemented this review of literature by survey, and thank you to all of you who participated in this survey. There were 2 reasons why we wanted to do it. Because what is published in the scientific literature may not necessarily reflect the real life patient. Because you get things published when they are different. Editors of journals and other people don't like things which are just the same because they tell you, well, we have seen these, so what's the point talking about it? So we wanted to do the survey just going to different populations and get an additional opinion whether the published cases truly represent this disorder. And that's why we also not targeted the specialists, but we targeted actually you, parents, and carers of these individuals to get a different second opinion. As you know, second opinion is quite important. So we have made it in different languages. That's a lot of work of Christy and everyone who helped here to get it into Italian and French in addition to English. We have about 186 contacts. The survey was open for a relatively short space of time. We are very happy to have a final set of 111. 126 people completed, but there were a few we had to exclude for different reasons. The advantage was also we didn't have only these heterozygous females, affected or unaffected, but we also have males, mosaic, affected, unaffected, and the germline also individuals who we call transmitting males. So these are the individuals who don't have this gene, and everybody was curious to see, well, it's such an important molecule, can we live without it, without having any consequences? Interestingly, we found almost identical results in terms of onset. As you can see, 12.2, there was 11.9, 10 months was the same. But we also found that in the males, it was not dramatically different. Also, there are small numbers, and the mean was shifted in males. With respect to other disorders like SCN1A or PRRT2, which are the frequent other epilepsy syndromes, the PCDH19 median are a little bit later. So the first, I guess, the summary points from this part was that the median onset in boys and girls, it's about the same. So from that perspective, they don't look dramatically different. The mean, it's different, but that can be also just due to small numbers at this stage of the males. So no major differences between boys and girls. Other useful outcome we saw today was that age of seizure onset, so the time the first cluster, it's probably a very strong predictor of the overall outcome. We heard that maybe a type of seizure may be a predictor, as well as the severity of the initial seizures, as we heard about status. So we use these graphs, and I'll show you a few of them. So this is all the individuals. They are divided on left-hand side and right-hand side. So these are individuals, about 47 or 50 here, who had onset after 12 months of age. And these individuals had first seizures before the 12th month of. We chose 12 months because that was the mean. And here you see the outcome. And when you see the line, like for example, this black one, that means that 2 individuals who had the onset of the first seizure after 12 months ended up with severe to profound intellectual disability. But you see proportionally, it's very small with respect to all of these individuals who had first seizure before age of 12 months. So there's many more of them. So clearly, the time on average, it may not apply to an individual patient, please don't take it that way, but it's a helpful information that the first seizure does predict the outcome. So this was based on the review. This is how it looks based on the survey. There is some overlap between the patient here and patients here, but generally these are different group of individuals. We have a lot of unpublished cases here, a lot of new mutations, and pretty much the outcome was almost identical. Now, something else which I put on this graph, I put this particular mutation, which is a missense change. And we had found 20 individuals different individuals from different families who are unrelated, as far as we know, who had exactly the same genetic diagnosis, but had very different outcomes. So this is the information that we cannot really correlate the type of mutation with the outcome, because there were individuals who really started to have seizures before 12 months and can be either moderate or can be very borderline. So even though I'm saying that before 12 months it's usually risky, but for these mutations you can have one or the other. You can even start after 12 months and you can still have moderate. But you see there is no line, green line, going from after 12 months to severe and profound, which is kind of interesting. Now, you say that's one mutation. So we found another one. So these are the 2 most recurrent PCDH19 mutations. So that means they are most frequently found in patients. And in this one, we found 30. So these 2 mutations just on their own explain 50 different individuals. And again, the purple, it goes here. And you see it's all over the place. So really by just having the DNA variant, it doesn't really help you to tell the parents what's going to happen. It's really the time of onset of seizures is better predictor, much better than the type of mutation. Now, what is also interesting, we have done this survey, and I'll show you a little bit other results, but you heard that there are a lot of neurobehavioral, neuropsychiatric problems, and that's why we have done the survey, because these are bona fide instruments other people measure these neurocognitive and neurobehavioral features. And we have also very large control populations, so we can be pretty sure where the baseline is and whatever it's above or below. So in this case, you can see again that the seizure onset really predicts the severity of other neurobehavioral features, not just cognitive outcomes. One way to look at it, these are individual patients, these are different scores. Earlier the onset of the seizure, higher the score, it means more severe outcome. As you get the seizures, first seizures later and later, you can see that the outcome is not as bad, but there are some who still score reasonably high. So again, early onset, not so good outcome later in the life. And these are individuals of different ages. So the summary from this is really the age at seizure onset is a good predictor of the cognitive and behavioral and general the neuropsychiatric outcome. So as part of the survey, we have done the genetics epilepsy measure, and even though I'm not talking about it, because we really argued with my colleagues, how do we measure the severity of epilepsy in girls clustering epilepsy? It's not trivial. Do we measure it by number of clusters, duration of clusters? It's very difficult, but when we look at it as a bulk, the severity of the seizures is a strong predictor of the outcome. But that's a statement, please take it with some grain of salt, because we didn't have a very good measure. We have done the social responsiveness and all the others, and we have done it on all individuals, not just the heterozygous females. These are overall the outcomes, and you see, so these are the total percentages, and this learning disability, executive function, autism spectrum, emotional problems. So you see that's about 45%, and the different colors talk about severe emotional problems, moderate or mild, but generally problems. So this is a summary of prosocial deficit, peer problems, hyperactivity. It's one of quite important, you go reaching up to 70%. And these are data against quite well-characterized controls. So there's a huge impact. It's not a seizure disorder, despite we have, it's in a title as girls clustering epilepsy. It is really just to aid the ascertainment of these cases, but this disorder ought to be understood as a lot more complex than just seizure disorder. This is another view. This little box indicates all those individuals. The blue are boys and the red are girls. This is the combined score. The blue box pretty much respond to the normal range. So all these individuals in this box do not differ in these measures from a matched population. So these, from that perspective, these are non-penetrant. But here what we're looking at was that the boys are not different from the girls. So the mosaic boys are not different. Because there was some speculation, maybe initially, that the boys might be protected, there's something about being a boy, etc. No, in this case, we can be quite confident that it is just a mix of the wild type and mutant cells, irrespective of the male or female gender. Or sex, if we talk about really XY chromosome. And that's the same also using different measures. Here, what is also interesting, so again, the green, the box green or blue, I don't know how you see it, is the normal values. And these two colors, the yellow one, it's non-penetrant females, and the blue one are transmitting males. So transmitting males are the knockout males. So these are the males who don't have PCDH19, don't have a gene, such an important gene evolutionary, and you would have seen it everywhere, yet they perform as knockouts perfectly fine as controls. This is also important because so far we have not had, there was some speculation that these transmitting males might be obsessive compulsive, they might have some neurocognitive, neuropsychiatric issues. It doesn't seem to be the case according to our study. They are by all means of these instruments not affected at all, unlike the unaffected females. So these affected males are same as heterozygous females, that's an important message, and the transmitting males are as much unaffected as unaffected females as far as non-penetrant females. Now, this was also mentioned, this is the work of my colleagues to some extent, Ingrid Scheffer, published recently, where, of course, we saw from the survey that there is another activity, there is something else happening when these girls, young women, become older, when they go through the menopause. At some point, even during later life, after puberty, 20s, 30s. So this team went back and looked at this number of females across many families, and they found 13% from 7 families had psychotic disorder. This is early days. It's something which Ingrid Scheffer, my colleague, noticed some time ago, and she followed up. So this is something we will need to pay attention to as this generation of young children grow up, and that will need to be managed. And you as parents, many of you talked to me, and I heard this clear and loud. We do need to do something about this and bring it to the attention of the services. And these are the numbers you would have heard, would have seen today already. So some individuals, and that is 1 in 5, may present with later onset, mean 21 years psychosis. The question is whether these individuals also will have increased frequency of seizures and other problems later in their life. Now, we didn't talk about this, but I saw some numbers, and we did see it, but this is very hard to measure, but about 1 in 5 individuals may present with some delay before their seizure onset. Now, this is not going to be trivial really to measure unless we're really going to do a newborn sequencing of almost everyone, and we'll ascertain these cases that way, or these will be cases born into families where they will know that they are carriers of PCDH19 variant from birth, and we'll be able to kind of test them before they get their first seizure. But there is some indication that proportion of individuals shows developmental issues before they first cluster. Now, what really bothered us and everyone, and it's not only PCDH19 epilepsy, but in many other disorders, is the fact that why the same DNA gives different outcome. In this case, we have male to female. We know these are transmitting males, and all of their girls have 80% chance to be affected because there is 80% penetrance. We also have many instances of unaffected mums or grandmoms who have daughters or granddaughters who are affected. Exactly the same genetic constitution. And if that was not enough, we have now 6 or more cases of identical monozygotic twins, by all means identical, as much as genetics tells us. We know that they are not truly identical, but for the purpose of the exercise they are, and they are discordant. So what's going on? If we were going to find this 10 years ago, nobody would believe us this condition is genetic, because by definition monozygotic twins should be almost presenting. So understanding of these non-penetrants is also important as much as understanding of the penetrants to really come to a solution for these girls. So these are the numbers. I use the prism to illustrate this, that one beam of light, in this case, it is the variant of PCDH19, come to the box of our body, our genetic background, and those who are fortunate escape, about 20%, and those who are less fortunate end up with these chances of these disorders or the combination of thereof. We know and heard, and I'm not going to talk about the function, but it's amazing what we learned about the function of these proteins, and we see in the cytoskeleton signaling, migration, regulation, in this case on the chromosome 16. It's a beautiful example of a microRNA, so another molecule which regulates PCDH19. And if you have more of that molecule, PCDH19 goes down. So in a way, PCDH19, it's not just a disorder of X chromosome, but it can be also very important for this chromosome 16 duplication disorder, because the molecule which controls PCDH19 sits there. So that, again, further enhances the importance of understanding this particular disorder. And then again, we found this very unexpected, we're still wondering what's going on, gene expression regulation by this molecule. And as you heard Sylvia very nicely about the link to GABA and more. However, there is one word of caution, and I could give you a lecture about this because I'm very interested in the mechanisms why this is happening. This group published some time ago that the loss of PCDH19 causes neural tube defects. That was about 2010. The same group 5 years later, using a different model, didn't see any of this. And I can give you many examples, Nature level, top scientific level publications, really offering totally opposing results. The difference is the technology. In this case, it's acute knockdown, so these cells live with the molecule, and suddenly we come with a tool and we'll cut it. While in this knockout, the cells start their life without that molecule, so they can chance to adapt. So the acute, even though may give us some information, we need to be careful how we take it. It may tell us the truth, but exaggerated truth. So we need to be really careful how we interpret. And then when we're going to test the drugs, we're going to consider both models and not perhaps just an acute model which is exaggerated, because then we're treating a different disorder. So this is kind of like a cancer in many cases, and this one is a genetic inherited disorder. So it's just the wrong technology, or it's telling us more about the biology. So we know the X chromosome inactivation, and Stefka and Jack showed you really beautiful pictures. Actually, Jack, the one of them, it reminded me of pizza. Maybe I'm getting hungry when you see the nice slices, the circle and the nicely cut pieces. I don't know, were they 12 pieces as well? I don't remember. So of course, X chromosome inactivation, we won't be able to fix. So how are we going to go about it? We heard about the treatment, and I'm not going to repeat that, but I'm going to repeat that there are very few actually good studies looking at this. And that's why we were crying to have more and probably better organized and more systematic. So this was the Higurashi, and Norimichi was here a couple of times when they found the corticosteroid therapy may help, but not over time. Then we heard about the stiripentol, and I felt like this is helping more girls than just this case as it started. And then we heard the German study from Lotte et al., where clobazam was probably the most efficient, but there were others. Now, we just submitted a very small study when we noticed that Keppra or levetiracetam showed efficacy. So these are the data which are, again, retrospective, but there were two cohorts. One, that was cohort of Lynette Sadlier in New Zealand and my colleague Ingrid Scheffer from Melbourne. They collected 17 females, and 13 of them became seizure-free for more than 12 months on Keppra, and 10 of them more than 24 months, which it's quite good when you look at the bottom line numbers. We looked into our survey with Christy, so this is where we asked this question to 111 individuals who responded, and in this case we identified 62 females in this range who trialed Keppra, and these are the number. 26 of these 62, 42% were seizure-free for 12 months, and 19, so 1 in 3 almost, seizure-free for 24 months. So from that perspective, it perhaps looks like at least something worth a try. But there are a lot of caveats to this. Of course, you see the age range, the irregularity of the clusters, and this was actually the Lotte's finding. So they had 15% of cases which were seizure-free. So it kind of replicated. These are just some patients. I haven't seen these seizure diaries. These are kindly provided by Lynette. When you see the clusters over first, second, third, fourth, fifth, and sixth year, and after Keppra was introduced, no seizures, flat. This is another case. This case obviously didn't respond as well. This one responded, or maybe the seizure frequency. But you see in this case, it just goes up and down, up and down. It's really not trivial to get a good feel for it unless you look over a longer period of time. We have not had a chance to look over a period of time. We only have this for 10 years. For many patients, 5 years or 4 years. This is a new disorder for services, research. So we probably need to appreciate that and incorporate it, but it shouldn't stop us to design better studies into the future of natural history. Again, we heard it, we need to have it. So the message is that there are no really good anti-epileptic drugs for the time being. And however, this is, if any time is good in history, this is the time. Generally, in gene therapy or therapeutic approaches worldwide, there's a huge activity. I was shown these pictures. They are probably a year or two old. So this is in oncology, immunology landscape, and they are concentric circles. So these ones in the middle are the approved drugs, and as you walk further outside, these are drugs in different stages of development. So this is CAR-T technology, or you heard, and oncology. And I'm trying to tell people that we have as much urgency in these neurodevelopmental disorders as there is urgency in oncology. We need to act early, as soon as possible, because once we miss the important developmental window where we can change the direction, we're probably not doing the best. So this is the gene therapy landscape, and you see there are a lot more closer to the center of the circle, and there are hematology, but also neurology, ophthalmology. It's quite amazing. So you heard previous talk, it is a good time, and I know of several other groups who are testing siRNA or antisense oligonucleotides in this space. Now, the issue is probably not there to what technology we use, but how we deliver it most efficiently to all these cells which are misbehaving. And that has been a problem now for many, many years, but perhaps there are better tricks, like you have seen about the adeno-associated viruses. So really, what is modifying the outcome? We know that to some extent it might be the variant and the X inactivation. I didn't talk about polygenic risk. So in a nutshell, maybe that's a not good comparison, but it really depends on which background or family background, DNA, this PCD H19 mutation lands. We know that this polygenic risk can add or take away up to 8% for moderate to severe intellectual disability. There is some stochasticity, and that's elegantly shown by work of Paul Thomas and our PhD student, and partly Stefka showed it, but I'll show you one picture after that. So in a way, when we look from the epigenetic perspective, because PCD H19 is probably an epigenetic disorder, it's the primary driver is epigenetics, that is a methylation which regulates gene expression differentially in different type of cells. So this is the canonical, very often shown picture of development. That's where we start, and we can have different valleys which way we go. That's a normal development, that's perhaps a mild development, or an alternative to normal development. And that happens across the lifespan. Under that landscape, of course, there's environment, development, there's genes, and we know for PCD H19 that we have a few genes we identified and confirmed, which are important. They are markers of PCD H19. So we get this complicated, almost epigenetic barcode underneath each individual, and that's why monozygotic twins can be different. Because even though they have same genes, this barcode, this landscape is going to be different because of the epigenetic driver influence. And this is, I guess, the example from Paul. I have all the pictures, so I don't have it in blue and green. I have it in red and green, which hopefully is visible to everyone. But these are, for purposes of this exercise, identical twins, yet they are not identical. And that can be the case also for human identical twins, that they will have a different barcode, which will determine. Now, we are trying to actually read this barcode to use it as a predictor. So I'm hoping that maybe in 2-3 years' time, we'll be able to test an individual at birth, see that they are PCD H19 positive, read their epigenetic barcode, and tell the parents, you don't need to worry because you are in the 20% non-penetrant, or we need to do something to manage it as soon as possible because you are likely going to be penetrant. I mean, that's the holy grail. That's what we would like to do because that's everybody will be asking for. So from my perspective, PCD H19 girls clustering epilepsy, it's a disorder of a lifespan. While we don't see obvious problems in early development before the children are born, not that I know, we know that the seizures can start early. They generally stop, but again, there are exceptions at around puberty. So this is really the time when things don't go well. Not so good news is that things happen later. The seizures may restart, and you have these other problems. And the question is there a delay even before the seizure onset? Now, in terms of the underlying problems, how this is driving it, so we can target it with our therapy or think about what to do, well, I probably naively, still is the hypothesis, think that maybe PCDH19 early on might be involved in regulation gene expression, because there is no need for cell-cell adhesion. Later on, we heard it's important for bundling of the cells, the migration, differentiation, axon or synapse function. What we didn't talk about, and it's very interesting, but we don't have a lot of experimental support, is that PCDH19 through oxytocin receptor might be crucial in GABA switch. So in a way, how the GABA receptors become more inhibitory rather than excitatory at a certain time. And who knows, maybe that's scrambled, because just simply dysregulates in oxytocin receptor, and that's one of the marker genes, causes major problems in GABA switch, perhaps. And then, of course, we saw nice studies from Milano about the synaptic function and from Jack. Whether there is still a role for regulation of transcription is possible, and you will hear about it probably tomorrow from Isabel about the role of the cytoplasmic domain, which we also looked into. And then there is the deficit, as we know, to response to stress. Now, this is something which is common for other epilepsies. The stress response pathways are broken. So I believe, I trust, once we're going to address PCDH19 epilepsy, it's going to have a lot broader impact to other similar types of epilepsies. When to intervene? I mean, early we can, but we don't know usually. We can do it at this level, at the decision landscape. Now, the question is, if we start intervening here, which might be at the level of the young child, well, how are we going to get the ball from here to its original position where it's supposed to be? Can we achieve it? Is it too late to intervene here? Do we need to intervene at that stage? And how to? These are fundamental questions where we have information from animal studies, but very little from human studies. So hopefully that will also happen, and we'll be able to understand not just how to deliver the tools to the right cells in the brain of these individuals, but also at what time. But we know fundamentally, earlier we do it, the better the outcome is. So last slide, I believe. We know that PCDH19 is disposable for males. We didn't know it. We are quite confident, even though the numbers are maybe still small, but the males can lose this particular molecule, important molecule, and they are as much as anyone else average in the society. It's possible also for females, I trust, based on the fact that the males and females are the same when they are affected, but we have not seen such a female with 2 mutations, one on each of her X chromosomes. The mosaicism has been now reconfirmed as the cause, and we know that mosaic males are technically indistinguishable from heterozygous females. So it's not the boy-girl disorder. It is a disorder of the cellular mosaicism, irrespective of the sex background. It is not just epilepsy. Seizure onset is a strong predictor, and that's a useful piece of information, because that can affect the way how the services are looking after these individuals. Whether delay precedes seizure onset is a question, but we also know very likely other comorbidities like developmental delay are probably independent outcomes of the same original cause and the problem. they may be exacerbated by very severe seizures, but they are likely independent outcomes. And it's drug resistant. And whether it's Keppra, clobazam, ganaxolone or others, well, hopefully we'll have answers. So that's where I will stop. I will say thank you, mostly my colleagues and collaborators, to all of you, to Francesca and to the foundation. Now, we also started this Human Disease Genes website when we'll be asking a lot of our clinical colleagues to actually all over the world to add their patient information, and you will instantly see graphs of that information, so visually you'll be able, and hopefully we'll grow this resource and see whether it's useful. This is not going to be as hard as doing registers. This will take literally like 5 or 10 minutes per patient, hopefully, and not dramatically more, but we'll see the statistics, at least in the basic indicators, over a long period of time, and we'll be able to communicate with all contributors. So it benefits patients as well as the clinical services in view of this disorder. So thank you very much for your attention.